And my name is Luther Beegle. I work at JPL with my co-authors are Everett Salas and Rohit Bhartiya. And this is the body in question, Enceladus, which is probably one of the more surprising astrobiology targets we've found in the solar system. In 2004, after Cassini went in orbit around Enceladus, in the south polar region of this body, they found an anomalously hot spot. This particular region has got a 20-degree increase in temperature among when you compare to regions right next to it. Basically, from this particular region, we see geysers and plumes raising from the surface of Enceladus. Enceladus itself is a very small body, and it's got a very high albedo. And one of the reasons why it's got a high albedo is the material that comes up from here can either go back, can continue to go into space, or it can land on the body. And the material here is mostly ice, as measured by the ion and neutral mass spec. If you look at this particular material coming off of here, it basically is concentrated in what are known as jets and in the general plume itself. These are Cassini observations. You can see the scattered light from the material. If you fly through with the ion and neutral mass spec, you get a nice chemical reading of mostly water with some other material in it. The material itself, once it escapes from Enceladus's gravity, the material that doesn't fall back onto the surface, basically makes up the diffuse E-ring. This material has been identified in orbit around Saturn since at least 1970. So the processes that are going on in Enceladus have been going on for quite some time, and probably much longer than the last 40 years. Now, as a card-carrying astrobiologist, as soon as you discover the fact there's probably a liquid water ocean underneath here, and you know that there's organic material on this surface, and you know there's an energy source, you immediately start thinking, wow, there's life. And what better place to go look for life than a place that's actually spewing the material out into space, because it makes everything much simpler. We don't have to land, we don't have to do any aquabots or anything like that. Let's just go pick up this material here. However, before we spend the $2 billion it would take to go out there, we have to ask ourselves a couple questions. And the first question is, what are we trying to measure? It turns out that there's only really one place in the universe we know that has life, and that's the Earth. So defining what life could possibly be in other places is very difficult to do. We would like to go have a search strategy that looks for material, that looks for life that wouldn't be like we would know it, but once again, that's difficult to do. So the second thing is we really need to understand it. If we're going to go look for a specific place, we need to know what the distribution of life is like on the Earth that kind of represents that particular place. Whoops. The second question we need to ask is, what's the platform that we're going to be measuring off of this? We need to understand the limitations of the hardware that we have and what that means in terms of a mission development scenario. So the assumptions that we make when we're going to, if we went to look for life on Enceladus, are basically that we would have to get organic material that's detectable in statistically significant quantities. A lot of people talk about measuring chirality as a determination of life. But if I only find one amino acid, I don't know what the D-Del ratio is of a singular amino acid. I also have the problem, if I have 100 amino acids, is it statistically significant to measure a D-Del ratio? At a certain point, I've got so little material that the statistics just don't work for you. And this basically affects any kind of search strategies you're looking for. Also, the second problem you have is that the number of biologically relevant or material that you find has got to be statistically significantly higher than what you're bringing with you, or what you find when you're out there from other sources, in this case, IDPs. In one paper last year, there were 14,000 different soluble chemical components found in the Murchison meteorite. We could probably make an argument that this is definitely not biotic, it's all abiotic. So if I'm going to do a statistical count of organic material to try to prove that there's life there, I have to make sure that I've got statistics, I've got enough material that I'm well above this particular limit. The second thing you really need to know is what was the exact bioload of your mission before launch. Terrestrial contamination is a major issue, especially when you're dealing with the very minute quantities that we're going to be talking about later. So Sorry for the misspelling here. I noticed it about 10 minutes ago. So what are some of the things we would look for in terms of life detection? Well, there's three basic forms of organic material as we think about it. One are the abiotically created or the prebiotic created, if you wish. What material that is considered a biosignature and material that is considered a biomarker. Biomarker is much more difficult level of proof than a biosignature. A biosignature itself can be amino acids, can be elemental abundances, can be a lot of different things. But a biomarker is a definitive proof that this sample was alive at one point. Protein is probably not a biomarker, it's more of a biosignature because it's very difficult to imagine how life could have started without abiotic protein formation in the first place. So one of the things we would want to measure is you'd want to measure the elemental abundance of any sample you had. Look at the CHNOPS in a sample. You take that sample and then you can do the elemental ratios, and I'm behind, so let me move ahead. You can look at chirality of amino acids. You can also go in and inventory amino acids. Over 70 amino acids have been detected in the Murchison meteorite. However, life only uses 20 amino acids. If you went in with a sample that was life and you measured, oh, there's 14 amino acids in this particular sample, they're different ratios, and in terrestrial life, that would be a good indication and probably go into the biomarker family. You could measure fatty acids. Life on the Earth tends to use C16 and C18 over the hydrothermal created fatty acids. And finally, DNA and proteins. The DNA, if you found a DNA with a different set of base pairs, you've got a biomarker. So what's in a cell? In the next couple of slides, anything written in red is something you should remember. Okay, your average individual cell on the Earth, this is a cartoon from one of my biology books, is about 2 microns in diameter. So this distance here would be 2 microns. Basically it consists of about 70% water and about 30% organic material. Of that organic material, if you dry the entire cell out, you're left with about 200 femtograms of material. Basically, depending on what different cell you have, you've got a different ratio of proteins, cell wall membranes, lipids, DNA, et cetera, but there's a basic breakout. And a good rule of thumb is this particular breakout here. So this is important for the amount of material we're going to be talking about later. So in each individual cell, there are about 2.3 million different protein molecules. Of those 2.3 million, there's approximately 2,000 to 3,000 different structures. Once again, the detailed inventory has not yet been done because you're talking about such small material. Of those proteins, if you break them up and you hydrolyze the peptide sequences, you're left with about 20 different amino acids that make up these proteins. Total DNA per cell is on the average of 2. There's a whole bunch of RNA you can see. Lipids are the same thing as a protein. About 22 million lipids in each individual cell. There happens to be about 1,000 different unique structures depending on what cell you're looking at and the like. And the rest of the cell from the organic material is the polysaccharides. A DNA molecule itself has approximately 3 times 10 to the 15 grams of material associated with it. Once again, depending on how many base pairs. So in the terrestrial environment, what do we actually see in seawater? Well, in 1998, McCarthy et al. Published these results from seawater. And what it basically shows is that the dissolved organic carbon in seawater that's unfiltered, this is all carbon, is only order of about 80 to 100 micromoles per liter. If you basically filter this out, remove all of the bugs, and just left with what's free amino acids and free organics, you're left with about 20 to 30, actually about 10 to 20 dissolved organic compounds in a sample. What this is basically if I take a 2 micron size ice particle, freeze it, of this ocean material, I'm left with about 0.3 to about 0.5 femtomoles of material per 2 micron size particle. If I have 1 million of these particular 2 micron size particles, which will become evident why 2 million in a minute, I'm basically up to about 3.4 nanomoles of material. Also, what he also noticed was if you looked at the D to L ratio of what the free amino acids were in seawater, you get a D to L ratio that's not necessarily 0. But with some material like the alanine, highly concentrated. So in these particular samples, what you find when you're looking at life in the Earth, what you find is that life basically is not necessarily homogeneous, it's inhomogeneous. If I look at a cell on a rock, I basically know that most of the carbon is contained inside these cells on this rock. If I look outside this cell, I'm not going to see very much carbon at all. If I look at an Antarctic sandstone, I'm also going to see material that's concentrated. Most of the organic material in an Antarctic sandstone all exists within the upper 1 centimeter of the material. If I look underneath here, I'm not going to see a whole lot of material. If I even go into the larger scales and I look at chlorophyll in the ocean, there's a 4-order of magnitude difference between the amount of chlorophyll here and the amount of chlorophyll here, which basically is only about 100 kilometers away. So if I want to go look for life, once again, I'm asking my 2 questions. What do we want to look for? We've previewed that. And what is the nature of the platform we can look for it? Well, on Enceladus, you can do one of 4 different things. You can look for the plume material. You can go in and look at the E-ring material, because once again, the plume material becomes the E-ring material. And I don't know what that's going to do to any individual molecules that are inside an ice particle. You can actually go back and do surface sampling, or you can basically do direct measurement. The last 2 of those things are basically much more expensive than just flying through the plume. For sample return, you have the advantage that you don't know what your actual limits of sensitivities are until 20 years later. So if I want to send something to Europa to do a sample, or Enceladus to do a sample return, I got 20 years of development of sample return instruments. In situ, you basically have to characterize everything now, and you've got to worry about all the sample processing. Getting to Enceladus is really not that hard. There are multiple mission scenarios that get you in, get you around with the same delta V that Cassini had, including one of them that basically lets you go into orbit around Saturn, visit Enceladus multiple times, and then come back for very little delta V. So, what's in the Enceladus plume? These are all best case scenarios for actually looking for life in the Enceladus plume. Their measurements have been about 350 kilograms per second of material emanating from these geysers. The vast majority of this a concentrated amount of this material is actually active in these jets. The speed of material coming from the plume is average about 600 meters per second. So at about 15 kilometers above the surface, and if I take a 10-kilometer wide disk, which is how big one of those jets are, and I assume that all the material is inside this 600-meter by 10-kilometer sized disk, what I'm left with is about 100 individual 2-micron-sized particles per cubic meter and an average particle density of about 20 micrograms per meter. So what does that get me if I fly through the plume? Well, if I have a 1 square meter collector, I get 200 milligrams of material, which is actually an awful lot. However, if I have a more reasonable 10-centimeter diameter collector, I'm down to 157 micrograms of material per pass in the Enceladus plume at the absolute best case scenario. This gives me about 1 million particles, and I get about 10 micrograms of material from the particles. So what I'm left with is, if I'm flying through a bacterial mat, I can assume that if I assume the same 70 to 30% water-carbon ratio, and of that 200 milligrams that I have, I get 60 milligrams of carbon-containing compounds, I get tens of milligrams of proteins, lipids, and DNA, and we're having a completely different conference 2 years from now. If I'm flying through a terrestrial ocean material, I'm left with less than a microgram of total material, and that all of that is organic, and that's all intertwined together. So at that point, you need to, you have a problem with cleanliness, and you have a problem with basically being able to do a quantitative analysis. You've got to have instruments that can measure things on the sub-picogram level. So conclusions, I'll wrap it up. Particle diameters tend to be on the same order of what a terrestrial cell would look like. In order to determine extant life through that in plume, you're going to have to do a quantitative, if you do a quantitative organic material, you're going to have to have a limited detection on the sub-nanogram level. You need to better understand inhomogeneities in terrestrial environments to basically understand what your probability of finding anything is. And the in situ analysis is going to be virtually impossible to do unless you go land on the surface and you're able to concentrate material. And with that, I thank the members of the organizing committee and everybody for listening to me talk.